That little brief overview was just to sort of give you an idea as to where I'm going with this comparison video. The first section of this comparison that I'd like to focus on is the light-dependent reactions of photosynthesis, which occur on the thylakoid membrane of a granum, as pictured here on screen, versus the electron transport chain of cellular respiration, which occurs on the cristae or the folds of the mitochondria on the screen. In photosynthesis, water is a reactant that is used to replenish the electrons lost by photosystem II after it has been struck by a photon. This splitting of water produces 2 hydrogen protons, 2 electrons, and half a molecule of oxygen in the thylakoid lumen. Electrons flow from photosystem II to photosystem I, and eventually to NADP+ reductase, where NADPH is formed. A high proton concentration develops in the thylakoid lumen from the splitting of water and the cytochrome Bf proton channel. These hydrogen protons are used by ATP synthase to drive the production of ATP from ADP and a P group. The following video clip provides a more detailed explanation of this process.
A photon of light hits a chlorophyll molecule surrounding the photosystem II complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem II, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem II are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient, similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created.
In comparison, the electron transport chain of cellular respiration begins at protein complex 1, NADH dehydrogenase. NADH is reduced to NAD+, and 2 electrons are shuttled through the phospholipid bilayer to complex 3. Complex 2 oxidizes FADH2 to FAD+. Complexes 1, 3, and 4 act as proton channels. A high proton concentration accumulates in the intermembrane space. It is at the 4th protein complex where water is formed from the addition of 2 hydrogen protons and 2 electrons to half a molecule of oxygen. Essentially, this is the reverse process of what occurs in photosynthesis. Here is a short video clip to provide a visual representation of the process. I also have a short clip that illustrates the process of chemiosmosis, or the production of ATP by ATP synthase. Keep in mind that chemiosmosis occurs in both photosynthesis and cellular respiration.
At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. Here we see ATP synthase. along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP.
Okay, so next I just have two quick short videos to show you. The first one is about the Calvin cycle, and the second is a description of the Krebs cycle, just so that you have an idea of what's going on and how these things work. And then I'm going to go into talking about how these two cycles are similar and how they are different.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
In the citric acid cycle, a 2-carbon compound reacts with a 4-carbon compound to produce the 6-carbon compound citric acid. In subsequent reactions, 2 carbons are lost as CO2, electrons are stored, and ATP is produced. By the end of the cycle, a 4-carbon compound remains, ready to react with another 2-carbon molecule and begin the cycle again.
The Krebs cycle is the cyclic stage of cellular respiration, which occurs in the fluid-filled matrix of the mitochondria. Meanwhile, the Calvin cycle of photosynthesis occurs in the stroma, outside of the thylakoid membrane. The stroma is also filled with an aqueous fluid. Let's talk about a couple of the key players we haven't mentioned very much yet. Firstly, carbon dioxide. As you saw in the short video clip, carbon dioxide is required for the first step of the Calvin cycle to occur, which is called carbon fixation. So in the Calvin cycle, carbon dioxide is a reactant. However, in cellular respiration, carbon dioxide is a waste product that is formed when the carbons from the original glucose molecule are released. The Calvin cycle also uses energy in a way that the Krebs cycle does not. The Calvin cycle in plants uses ATP and NADPH to store energy in the form of a glucose molecule after 6 turns of the cycle, while the Krebs cycle breaks down the pyruvate that originated from glucose to make ATP, NADH, and FADH2. So while plants are making and storing energy from smaller building blocks, heterotrophs like you and me are breaking down the stored energy in large molecules. This is why autotrophs are anabolic and heterotrophs are catabolic. As you have learned from this video, the processes of cellular respiration and photosynthesis are symbiotic because they depend upon one another. Animals depend on plants to produce oxygen and glucose, while plants rely on animals and other organisms to produce carbon dioxide. Evolutionarily speaking, scientists believe that the first cells on Earth were capable of neither photosynthesis nor cellular respiration. However, photosynthesis had to have developed before cellular respiration, given the abundance of oxygen in the Earth's atmosphere. Approximately 20% of Earth's atmosphere is composed of oxygen. This translates to billions of years of photosynthesis.